So Dr.
Dr.
Sundaresan will be the next speaker, and he's going to talk on mitochondria and wound healing. Is the topic, and we are running behind schedule quite a lot, and therefore requesting Dr. Sundaresan and Dr. Tandon to give a short talk, so that by 4:45 we'll wind up and leave this hall for the next session. Thank you.
Okay, thank you, Dr. Kumar. So I will, lack of time, I briefly emphasize my point. So I'm going to emphasize 3 points. One is what we did for the past 25 years related to corneal genetics. Second one is what we did for the mitochondrial research for the past 25 years with the various eye diseases, and what is the interaction between the mitochondria and wound healing. So concern with the cornea, you know the endothelial dystrophies, there are 3 types, CHED, Fuchs, and then PPCD. So in 2006, till 2006, the candidate gene is not identified. Our research group, in collaboration with the Singapore Research Institute, we identified the candidate gene for CHED2, that is the autosomal recessive disease. The candidate gene SLC411 is present in the 20th chromosome. So we did all the expression studies, and then we cloned the gene, and then we expressed the gene, and then we did the cell culture model, Western blot, and everything. We proved, yes, this is the candidate gene for CHED2. This data was published in Nature Genetics. I'm one of the first author in this paper. Second one, concerned with the Fuchs endothelial dystrophy. There are a lot of candidate genes are there, but there is again SLC411 is a candidate gene. There is a crosstalk, but the collagen gene, collagen 8 is very, very important gene for this particular disease. So we entirely screened, and then now we have a spectrum of genetic variation for Fuchs endothelial dystrophy. And then macular corneal dystrophy. This is very recently our publication from Molecular Vision. We screened more than 130 macular corneal dystrophy patients. And then concerned within the Indian population, there are 3 types of macular corneal dystrophy. I mean, the types, phenotypes. One is the type 1, 1A, and then 2. So in concern with the Indian population, the type 1 is very, very common. So that's what we reported through immunohistochemistry. And then concern with the mitochondria, because I'm going to correlate with what is the role of mitochondria in cornea. So concern with the cornea, we extensively studied what is the interaction between the mitochondria and the nuclear genes. So you have to understand the nuclear gene is separate and the mitochondrial gene is separate. In the mitochondrial genome, there are 13 genes are there. And then the 13 genes is interacting with the nuclear genome. We have 19,000 genes, and then depending upon the disease, there is a crosstalk. Concerned with the first paper, primary open-angle glaucoma. There are primary open-angle glaucoma, and we studied what exactly the mitochondrial crosstalk with the mitochondria and primary open-angle glaucoma. We published in a very high impact journal. In addition, the right-hand side, so we taken the cadaver eyes, and then we segregated control diabetic and diabetic retinopathy cadavers. From that, we take the neural retina, and then we studied what is the exact role of mitochondria in the mitochondrial pathogenesis. That also is the entire more than 32 pages publication is published in Mitochondria. And then concerned with the mitochondria for the past 15 years, concerned with the Leber hereditary optic neuropathy. So what exactly the primary mutation, what exactly the secondary mutation, what is the crosstalk between the nuclear genome and mitochondrial genome, and hospital-based studies. Because whenever we refer, we are quoting only the Western population. Concerned with the LHO and Leber hereditary optic neuropathy, 95% of the patient positive for primary mutation. In our case, is different. So that's what we are looking the entire what is the crosstalk between the mitochondrial genome and nuclear genome, where exactly the nuclear genome is interacted, either in the complex one or in the mitochondrial dynamics. We extensively studied for the past 15 years. All these papers are published. Please, go through our publications. So concerned with the cornea, concerned with this particular topic, wound healing. So recently we made a study design, and then we are preparing a study protocol or study design. That time I came across this very good paper. It is published in IOVS very recently in 2024 from Israel. So here I think I don't want to go line by line. The content is you take the corneal cells, the human corneal cells, and then you isolate the mitochondria from ARP cell line, that is the retinal pigment epithelium cell line. From that, you can isolate the mitochondria and then incubate the mitochondria with the corneal cells. When it's incubated, it means the exogenous mitochondria, it is entered into the cell, and then once it is entered into the cell, it increases the ATP production as well as the membrane permeability. So due to this, the wound healing process is very high. So that is the point. So here, in addition, they made an animal model in the sense they made an alkali burn in mouse model, just exactly alkali model, and then in the mouse model, and then they isolated the mitochondria from the liver of the mouse, and then now they have the mitochondria of the mouse from the liver, and then they added topically, and then they are checking how it is the wound healing, and then the the corneal thickness, they are measuring that one. You can see here in the figure 1, the green one is nothing but a kind of mitochondria is inside the RPE cells. From that, we can able to isolate, and then you incubate with the epithelial cells. You can see the green one, the exogenous mitochondria is entering into the cells, and then the green signal, you can see it is entered into the mitochondria. The control, it is not entering. So that is the flash slide it is representing here. In addition, this is the wound healing, the control, and then mitochondria. This is nothing but a kind of corneal cells. You can plate it in the 0 hour, 24 hours. In the control group, there is no proper wound healing. In the scratch model, the mitochondria in the sense you have to apply the mitochondria, whatever you isolated from the liver cells, and then you can see the wound healing closure percentage. You can see the second histogram. The mitochondria, the percentage is very high. It means the wound healing process is very high when you incubate with the mitochondria. And then concerned with the proliferation. The proliferation of the cells also is increasing when it's incubated with the mitochondria. You can see the difference between the control and the mitochondria. Obviously, the proliferation is very good, meaning it is enhancing the wound healing. Then this is again, it is in the animal model, how the mitochondria is entered into the cornea. That is the kind of representation. They have put a specific marker to detect the mitochondria. You can see it in the yellow color, and the green one is the nucleus. Here, as I told you in the animal model, just alkali burn, you make alkali burn, and then you add the mitochondria topically, and then you can observe day 1, 2, 3, 4. In the first panel, the control, there is no clarity, and then you can, within the 4 days, the corneal is recovery, promotes the corneal recovery. And then the wound healing, the graph, you can see the same thing representation within the day 4. When you add mitochondria, I think the wound healing process is perfect. So, and then they are measuring the corneal thickness. In the control, the corneal thickness is not that much reduced. When you add the mitochondria, the corneal thickness is reduced. So based on this, the wound healing, when you add mitochondria, is playing a major role in the sense it is once it is entered into the system, the cell, it is increasing the ATP production as well as the cell permeability. Based on that, the wound healing process is perfect. So I think this is the conclusion. I don't read the same thing. I already emphasized. And then the conclusion is the mitochondria promotes the corneal re-epithelialization and wound healing. The augmentation of the mitochondrial levels via mitochondrial transplantation may serve as an effective treatment for including the repair of corneal epithelial defects. So this is the points I would like to emphasize. Concerned with the gene therapy for cornea, so now as I mentioned in the very beginning, so right now people are adopting the gene editing technology, either the CRISPR-Cas and like that. So there are so many techniques are there, RNA therapies, stem cell therapies like that. In Australia right now, recently I think there is a paper published in JAMA. Just they're adopting the RNA therapies for Fuchs endothelial dystrophies. If during the when you treat with the RNA therapies, definitely the progress is very good when compared to the control. That's what they're emphasizing. That kind of research is going on. So with this, I'll stop. Thank you very much for your attention.
Thank you, Professor